Welcome, medical students, to today's histology lecture on the topic of cardiac histology. In this comprehensive script, we will delve into the intricate details of the histological structure and function of the heart. From the atria to the ventricles, from the atrioventricular valves to the epicardium, we will explore the various components that make up this vital organ. So, let's begin our journey into the fascinating world of cardiac histology. The heart consists of 4 chambers, the atria being 2 of them. The atria are thin-walled structures located at the superior portion of the heart. Histologically, the atria are characterized by a layer of endothelium, known as the endocardium, which lines the inner surface of the chamber. The endocardium consists of a simple squamous epithelium, providing a smooth surface for blood flow. Beneath the endocardium lies a subendocardial layer composed of connective tissue and small blood vessels. Moving on to the ventricles, these are the lower chambers of the heart responsible for pumping blood to the lungs and the rest of the body. Similar to the atria, the ventricles are lined with the endocardium. However, the ventricular walls are much thicker due to the increased contractile force required for pumping blood. The subendocardial layer in the ventricles contains numerous blood vessels, allowing for efficient oxygen and nutrient delivery to the myocardium. Now let's focus on the atrioventricular, AV, valves. These valves, namely the tricuspid valve on the right side and the bicuspid, also known as the mitral valve on the left side, separate the atria from the ventricles. Histologically, the AV valves consist of folds of endocardium reinforced with connective tissue. These folds, called valve leaflets, are anchored by fibrous structures known as chordae tendineae, which prevent the valve leaflets from prolapsing into the atria during ventricular contraction. Next, we move on to the semilunar valves. These valves, namely the pulmonary and aortic valves, are located at the entrances of the pulmonary artery and aorta, respectively. Histologically, the semilunar valves consist of 3 leaflets composed of fibrous connective tissue covered by endothelium. They are designed to prevent backflow of blood into the ventricles once contraction ceases. Now, let's explore the histology of cardiac muscle tissue. Cardiac muscle cells, also known as cardiomyocytes, are responsible for the coordinated contraction of the heart. Microscopically, cardiac muscle cells exhibit a striated appearance due to the arrangement of contractile proteins within their cytoplasm. These cells are branched, forming interconnections with neighboring cells to ensure synchronized contraction. A unique feature of cardiac muscle tissue is the presence of intercalated discs. Intercalated discs are specialized junctions that mediate cell-to-cell communication and mechanical coupling between adjacent cardiomyocytes. These discs contain desmosomes, which provide structural support, and gap junctions, which allow for the passage of ions and small molecules, facilitating electrical signaling between cells. Let's now shift our attention to the endocardium, which lines the inner surface of the heart chambers. Histologically, the endocardium is composed of a layer of endothelial cells supported by a thin layer of connective tissue known as the subendocardium. The endocardium serves as a protective barrier and helps maintain a smooth environment for blood flow, preventing clot formation and minimizing friction. Moving on to the outer layer of the heart, we encounter the epicardium. The epicardium, also called the visceral layer of the serous pericardium, is composed of a thin layer of connective tissue covered by a mesothelium. This layer helps reduce friction between the heart and the surrounding structures during contraction and relaxation. Let's now zoom in on the right atrium. Histologically, the right atrium exhibits similar features to the other atria, including the presence of the endocardium lining its inner surface. In addition, the right atrium contains specialized structures such as the sinoatrial, SA, node, also known as the pacemaker of the heart, which is responsible for initiating the electrical impulses that regulate heart rate. Shifting our focus to the left atrium, this chamber also possesses the characteristic features of the atria, including the endocardium lining. It receives oxygenated blood from the lungs and pumps it into the left ventricle. The left atrium plays a crucial role in maintaining circulation and ensuring efficient oxygenation of tissues. Let's now venture into the right ventricle. Histologically, the right ventricle exhibits a thicker myocardial layer compared to the atria, allowing for more forceful contraction to pump blood into the pulmonary artery. The papillary muscles, extensions of the ventricular myocardium, are connected to the chordae tendineae, enhancing the structural integrity of the tricuspid valve and preventing valve leakage during ventricular contraction. Lastly, we explore the left ventricle, known for its powerful contractions to propel oxygenated blood into the systemic circulation. The left ventricle has the thickest myocardial layer among all the heart chambers, ensuring efficient delivery of oxygen and nutrients to the rest of the body. The bicuspid valve separates the left atrium from the left ventricle, preventing backflow of blood during ventricular contraction. In conclusion, the histology of the heart exhibits remarkable complexity and precision in its structure and function. From the architectural arrangement of cardiac muscle cells to the intricacies of valve histology, each component plays a critical role in the efficient pumping of blood throughout the body. Understanding the histological features of the heart provides valuable insights into its normal function and provides a foundation for comprehending the pathological changes that occur in various cardiac diseases.